Okay, so in just a moment, Jeremy's going to take off with an extended assembly demo. It looks like everything is sharing correctly. If you are not interested in this, you may want to wander off to another space because there's going to be a lot of talking for the next 2 hours about a topic. Just letting him set things up right now. Any questions before we get started? Can you paste that into the Slack?
Yes, I can paste this into the Slack. That'll be much easier. No. Bunch of material. Okay, I'll give everyone a little while to download and unzip this. Can everyone hear me okay? Can I get a show of hands how many people have actually done some kind of assembly before? This is basically just a brief introduction to the kind of input data that we get, so sequencing read data, a brief description of what each program does, and then we'll have a look at the output data. Okay. How about if I lean forward like this? Is that better? Okay, I'll try and lean forward when I talk into the mic, but I need my hands, so I don't want to hold the microphone the whole time. Once everybody's got the zipped file and unzipped it and they're in the folder, just raise your hand for a few seconds so I can see where we're at. Okay, so the situation here is that we have error-corrected PacBio reads in FASTQ format from sugarcane mitochondria. And if you look at the file, this is reads.fq. This is just a small subset, so you can probably actually open this in a text document. We'll open it up and have a look at what it looks like. Maybe trying to open it was a mistake. Okay, maybe you guys can try and open it on your computers and see if that works. Otherwise, maybe we probably should try and open it. My computer seems to be having trouble running the screen share and doing all of this stuff at the same time. Okay, so if you actually did manage to open it and this is big enough, it looks like this. So we have the header of the read here. We have just a ton of A, C, T, and G. Then there's a plus, and underneath is the quality of each base, although it's not showing up properly in this. And we have a huge number of those. Oh yeah, that'll be much better. Is this better? Yeah, that's much better. Okay, so there's 2,700 lines in reads.fq, so it's not particularly large. Anyway, the point of the exercise is to see what difference is made between having long read versus short read data. So the error rate of the short read data should be roughly equal to Illumina, because these reads have been error corrected using 40x of the short reads from PacBio. So the difference in the assembly quality that we get is due to the assembler method used and the read length. Okay, so the first one, I have split these reads using the code in R code. So you don't need to follow along with this bit because R is a language, so it has a fairly steep learning curve. So I split, where did I do that? Here. So I just took a subseq of all of the reads, making them 150 base pairs long. So we have paired end data of 150 base pairs on each side with a 500 base pair insert. Okay, so to run the code, it looks like this, and you essentially just paste that into a terminal like this. So it's a command line program. Here we've set the k-mer size to 31. Use it, the reads are split reads.fa, and the output, we're telling the program to output everything into empty array. So if we go back to that folder, this is the empty array output folder. And so if you look in there, this is all of the output data that you get from Ray. So the thing that we're most interested in is the contigs, which are in the file called contigs.faster. And here you can see that here, 667 KB, so this should be a lot easier to open in a word processor. Okay, this one actually works. So here you can see it just labels each contig with a number. It tells you how many bases are in that contig. And you get, how many pages is this? 199 pages of A, C, T, and G. So remember, this is just the mitochondria. So this is one of the smallest genomes that you'd be assembling. If you're assembling an actual entire genome, you get a very large output file. Thousands of pages and several megabases of file size. Okay, so the question for this file is how many contigs are there and what length are they? So if you tried to do this by hand, you'd have a lot of trouble. It'd take a very long time. So I do it using this language called R, which is essentially a statistical analysis language. It was designed for data analysis. Okay, so I need to use these two libraries. And I'm going to read the contigs.faster file into my computer's memory. So because I use R, Because I use R, I have a computer that has a large amount of memory. So this has 32 gig. Okay, so now I have a large DNA string set, and it says there are 786 elements. That means there are 786 contigs. And you can see a representation of them here. The program just gives them assigns them a name in increasing numerical order based on when the program assembled that contig. So here I'm making a data frame that has the name of the contig with the length of each. And so that looks like this. So here's the contig names, and this is the length of each contig. So if I take the summary of that, the shortest contig is 123 bases. The median contig length is 202 bases, and the largest contig is 19,869 base pairs. Okay, so if you remember back from the first talk, Ray is one of the hybrid assemblers. So it uses a De Bruin graph, but instead of discarding the reads, it keeps them and then maps the contigs back to there. So it also outputs a scaffolds.faster file. So in this case, we didn't actually use any paired end reads. So it's the same as the contigs faster file. So the next step is to try blasting some of the contigs against the NT database. So that's the database in NCBI that includes all of the nucleotide information that we've ever gathered. And we can do this to see what sequence they're from. So if you have that file open, we can try and do just a web blast of the first one, for example. Although typically you do this on your own cluster, you wouldn't try and do it in the web server. So the point of doing the blast here is that we're expecting to get mitochondrial DNA. But mitochondrial DNA has a tendency to copy and then get incorporated into the nuclear genome. So although we're trying to assemble the mitochondria, there will be nuclear contigs that have been assembled in here. So we need to identify those ones and exclude it from further scaffolding steps. There could also be contamination in the samples, and so these would blast to a different species, and then you could exclude those contigs. So in this case, the top blast is Saccharum hybrid cultivar chloroplast genome. So the chloroplast, the mitochondria, and the nuclear genome all have DNA that jumps between them. So the mitochondria has maybe 10 to 30% chloroplast, and the nuclear genome has about between 20 to 50% of the complete chloroplast and mitochondria DNA that's been incorporated into it. So if we want to assemble the mitochondrial genome, we need to exclude contigs that are completely chloroplast and contigs that are identified as nuclear. So in the practical, I usually get the students to blast a handful, but we probably don't need to go through the tedium of blasting a large amount. So we get some chloroplast, mostly mitochondria. And some nuclear contigs. So we can make a blast database and blast the contigs against each other. And so this is using command line blast. And so the code here is make blast db. The input is contigs.faster, and the db type is nucleotide. And have I done that here? Yeah, so that's in this folder, contigs bdb, and that gives these output files. We can then do a nucleotide blast of the contigs against themselves, and then the output from that is the file called contigs versus contigs. I've set the e-value to 1e to the minus 12, and the out format is 6, which tells BLAST to give us a tabulated output of all of the matches. So that's this file. And so here we have the query, the match identity, the length. This is mismatches, gaps, query start, query end, match start, match end, the e-value, and the score. So here we can see that contig 0 matches perfectly to itself, as we'd expect. There's also what appears to be an inverted repeat. No, it's a palindrome. So the sequence this way is the same as the sequence that way. And we can see that contig 0 has several smaller matches to each of these other contigs. So generally in the range of 50 to 250 base pairs. So these are small repeat sequences that are occurring in both places in the contig. Okay, so this BLAST output allows you to identify contigs that reverse complement other contigs, or contigs that are partial sequences of larger contigs. In some cases, it allows you to identify contigs that have overlapping ends, allowing for larger contigs to be generated. So the question here is, are there many overlaps and why? So to answer that, I use R. So I've read in the table. I'll give the columns their proper names. Okay, ID, match, identity. And since we need to know if the match is at the start or an end, we need to know how big the contigs are. So what I just did there was to add the contig length of the match and the contig length of the query. And so now we can see here the match is this contig, it's 2 KB, and the match occurs at the end of this contig. And it occurs at the start of this contig. So that means that these two contigs can probably be joined together. And the reason that the programmers split them up is because there's going to be more than one join. So if I drew that, if I can hear that. So we'd have a case of this being contig 0, and then it matches to this contig, but it also matches to another one. And so the programmers split the contigs up so that you get the large contig, you get this contig with a small overlap. Okay, so the next thing that we'd probably like to know, to answer this question, are there many overlaps? You need to essentially go through this entire table, which is 3,742 rows, and identify how many overlaps there are. So again, this is not a thing that you do by hand. So this is why I use the language R. So the first thing is you'd want to get overlaps that are large. So this is choosing alignment lengths that are greater than or equal to 500 bases. And so that's cut that file down from 3,700 rows to 168. We want to exclude cases where the ID contig and the match contig are the same. So that's cut the file down to 14 rows, and that's a size that we can actually look at. And then it's just a matter of reading through to see if we have large overlaps at the start and end of contigs. And so here, this contig has an overlap with this one at the start, but for the match, the overlap is in the middle. So this suggests that the shorter contig is a repeat contig. So we can probably ignore those ones. And if we go through the entire table, you see that there's not a large number of very useful overlaps. So the answer here is not many. And the reason is that the repeats are larger than the read lengths. So we've already extracted as much information out of read length this size that we can. So another thing that we can do is to blast the reads back against the contigs and identify which contigs join to which. And so this is a very similar approach, and it gives you an idea of how you can piece the contigs back together. And so here's the code to do that. So we're using a nucleotide blast, the contigs as the database, and this time the query is the split reads.faster, and the output is reads versus contigs. Which is this file here. It's a bit larger, so I won't try and open that in a text document. So the output of this is almost 50,000 rows. And again, it's just a blast table output. Okay, so we need to merge in the contig lengths. And now we can see where each of the reads map to within the contig and how big each of the contigs are. So here we're interested in reads that match to the start or the end of a contig. So we can sort that table to select for entries where the ID sequence start is less than 50, or the query start, and the query start is greater than 200, or where the read end is greater than or equal to the match length minus 50. So we're trying to select for reads that have a 50 base pair overlap to the start or end of a contig. And we get 9,602 of those. So here we can see this small portion of the this contig matches to the start of this read. And then you'd basically just have to go through that table and try and piece the contigs together as best that you could. So the idea here is to find reads that partially map to the start or end of one contig, and then partially map to the start or end of another contig, allowing you to join those two contigs back together. And so if we went through all of that, we'd find some. Okay, and the next part of the practical wheel is realize that this is really hard and not something that you do by hand. And so what we've just done is essentially what scaffolding programs like SSpace are for. But these programs need paired end read data. So we have 500 base pair reads that we can make paired end read data from by taking the first 150 base pairs of all of the reads and the reverse complement of the last 150 base pairs, giving an insert size of 500 bases. So here you can see we have read 1, and then this is read 2, and we know that there's about 200 bases of unknown sequence in between. And I made that using this code, and then output the file to read split pedend1.fa and read split pedend2.fa. And I think that's in here. Again, so each file has the same read name in the same order as each other. So when the program has both of the files, it knows that read 1 from file 1 matches to read 1 from file 2. So it's important that if you're filtering the reads, that you filter both files equally. So you can't say remove a low quality read from file 1 and leave the matching read in file 2. It'll cause a problem when you later on when you try and put that into programs. Okay, so the code to run SSpace is this. We're telling it to use contigs.faster, and I made this so long ago I can't remember what these options actually do. But if you do use SSpace in the future, you can read through the manual and it explains what each of these options are for. And I think we have an SSpace output in mtarray. In SC also. This is the output of running SSpace. And the scaffolds are in here. Again, it's just a text document. Okay, so the scaffolds are in this file, and it gives a list of which contigs form which scaffold in the sc all.final.evidence. So the question here is, how much of an improvement was there? And I don't think I have that in my R code. But we can, to identify how many scaffolds there are, we can just search for a greater than symbol, which always starts the name. And this says there are 749 scaffolds. So if we go back to how many contigs there were 786 contigs. So 786 contigs have been collapsed to 749 scaffolds. Which is not much of an improvement. But that's to be expected because we have short read data. And so if you remember from the lecture, the problem of short read data is that the short reads are not long enough to span repeat sequences. And so every time there's a repeat sequence larger than the read, it generates three separate contigs. So usually scaffolding is supposed to be done with long-range paired end data, such as 4 kb, 8 kb, or 20 kb. So we don't expect much just by reusing the 500 base pair reads. Again, so the takeaway message from this is that genome assembly using short reads doesn't give you a very polished assembly. And so if this was the only data that you had, this is as good as a genome as you could get, 749 contigs. So it's not a very polished assembly. Okay, now we'll move on to the long PacBio reads, which we're going to assemble using the program Canu. So to run Canu, we use the following code. So assemble, where this is what we're going to call the output file. We're expecting a genome size of 1 meg. PacBio has a large error rate, so we have to tell Canu what the expected error rate is. And we're using reads.faster as the input with 24 CPUs. So the output of this is in the folder called mtcanu, and the contigs are in the file called mido25, which is what we named it, consensus.faster. They're in here. And this. Okay, and the first thing that we can see is that the first contig is 334.9 kb. So if you compare that back to the largest contig from Ray, which was 19 kb, this is a heck of a lot better. Okay, so how many contigs are there and what length are they? So this one's a bit easier to answer. I'll let you guys try and figure out the answer. Whoever gets it first can yell it out. So remember that the names always start with this greater than symbol. I think somebody said 5. Yeah. Okay, so if I read in the mitocontigs, there are 5 of them. So immediately we can see that the assembly using long reads is a heck of a lot better. So the largest is 334 kb and the shortest is 28 kb. So the next step now is to try and figure out if these are all mitochondrial DNA, if some of them are nuclear DNA, or if some of them are chloroplast DNA. Again, so we'd do the same thing as we did before, and we'd blast them against the nucleotide database to see what sequence they are from. So the trick to copy this is to search for the greater than symbol, identify the end of the contig that you want, and then just hold shift to select the whole thing. Although I think 334 kb is too large to input into a web blast, but we can give it a go. I think it gave me a problem last time I tried. So if some of you want to try and grab the other 4 contigs, maybe we can get some result from all of these. I'm just going to grab some water. NCBI might be having some traffic. It usually doesn't take this long. Okay, the last one blasts to genomic sequence, sugarcane genomic sequence. Did anyone blast 4, 11, or 13 and get a result? This is better done in smaller groups. It can be more interactive. So this one blasts to genomic as well. This one blasts to mitochondria. And one of these blasts to mitochondria and the other one blasts to chloroplast, but I can't remember which. Did anyone blast contigs 4 or 11? Okay, 4 is chloroplast, excellent. Okay, so the result from this is 2 mitochondrial contigs, a chloroplast contig, and then 2 genomic contigs. So mitochondria, we expect to get a single cyclic sequence, which means that we expect these contig 0 and contig 11 to somehow link up. So the next step would be to make a blast database of these contigs and then blast them against each other. So we'd do that with this code. So the input file is the contigs with the database type as nucleotide. And then we'd blast the contigs against the contig database file. And so the output from here is mito versus mito, which is here in the empty Canoe folder. So when we read that in, there's 545 blast matches. And so then it's good to have a table to say, so that we can merge in how large the query and the reference sequences are. Okay, so now we have that table that we had before with the query sequence as ID, the match sequence, and then we can see the length of each. Okay, so we look for cases where ID doesn't equal match. there's 300 of those. Mostly it's internal sequence. So in most cases, what we've identified here is repeat sequence. And so these are the repeat sequences that were causing us to get so many contigs when we had short reads. And so here, the alignment length. So these repeat sequences are 5,000 bases. We have one that's 2,400, one that's 1,600. And so the short reads being 500 bases is not enough to span these repeat sequences. So these were coming out as individual contigs when we were assembling with short reads. Okay, and so the question here is, are there many overlaps? And the answer is, there are a lot of overlaps. And the reason is because of repeat sequences. So the next question is, what can you see about the start and end of mitochondrial contigs? And so for this, we can look at mito versus mito. And it says here to remember that this is an OLC algorithm. So if you remember back from the lecture, OLC gives overlaps at repeat sequences, because what OLC does is stitch reads together. And so when a read goes into a repeat sequence that's too long for a long read to span, then the start of that repeat sequence gets added to the end of the resulting contig from there. So if we let's look at the first 5 kilobases and the last 5 kilobases of each contig. That's not giving anything. Okay. What we can also do is blast the long reads against the contigs to find reads that map partially to the start or to the end of a contig. So if we have cyclic contigs, we should expect a read that maps about half to the start and then about half to the end of the contig, meaning that that contig is cyclic. So here's the code for that. Using nucleotide blast again. Here the database is the contigs, and the query is reads that are 15 KB or larger. So 15 KB or larger reads is here, So this is the subset of the input reads that are at least 15 kilobases. And I think this is the code that I used to make that. So this is just reading in all of the reads, 675 of them. And there are 296 reads that are at least 15 kilobases. So the largest of those reads is 39 kilobases. The median length is 25 kilobases, and the shortest is a little bit over 15 kb. Okay, so the output of that blast is in MT Canoe, and it reads 15 kb versus MITRE. Gives 5,269 blast results. Okay, so here we have the ID of the read, the config that it matches to, and where it matches to on that config. Now we need to know how big that read is, how big the config is. And then we can figure out whether the read matches to the start or the end of a config. Okay, so here we're subsetting that table so that we get reads at the start or the end. That gives 71 results. So as you've probably noticed, most of genomics is not a graphical representation. So a lot of this stuff you have to do in your head, and then you code it, and you get the output, and you hope that what you coded gave the output that you expected it to. Okay, so if we look at the first read, it matches to config 0. The seems to be the end of this read. So from about 7 kb up to the end, which is 17 kb, so we can see that the read length here is 17. The last sequence that matches is the end here. It maps to the start of config 0. So the next question would be, where does the rest of that read map to? So we need to go back to the full table. And get all of the matches for this read. Okay, so these are all of the matches for this read. You can see a lot of small alignments. What's this, like 40 to 90 base pairs? So these are repeat sequences. So we can safely ignore those. In fact, let's subset them away. Okay, so all of those smaller matches are gone. We can see that this read in its entirety matches to somewhere in the middle of contig 0, and a portion of this read matches to the start of contig 0, which suggests that contig 0 has a cyclization point at the start and in the middle. Does anybody have any ideas what that might mean? So it turns out that the mitochondrial genome actually has two completely separate cyclic, I guess, chromosomes, although you don't usually refer to a mitochondria as a chromosome, but it has two completely separate cyclic sequences. And so in the case of this overlap, it has a repeat sequence in the middle and at the start. Let me see. I'll go to the paper and show you a picture of what it looks like. Where did I put that one? Okay, so it actually looks like this. So this is what we're saying is contig 0. And this is contig 11. And although there's a few short repeats that are shared between these, there's almost zero sequence overlap. So they exist within the sugarcane genome as two completely separate pieces of DNA, which is very unusual for mitochondria, although it seems to be a lot more common in plants. In animals, there's only ever one mitochondrial cyclic sequence, and the mitochondria in animals is a lot smaller than the mitochondria in plants. Again, so the answer from this is that output from Canu pretty much was already almost perfectly polished. I think I ended up having to cut a few kilobases off the end of one of the contigs, but otherwise the output is exactly what we got. Yeah, this contig 0 is too large to blast. That didn't work. Okay, that's the end of the assembly practical. Travis was telling me that some of you are interested in doing reference-based assembly in your project. If any of you are interested, I can go through how you'd go about validating the output of that. It's very not hands-on. It's kind of just me running through some code and explaining it. So anybody that wants to walk out now, go ahead. Anybody that wants to stay, I'll go through what I'm doing in a species of mangrove. Okay, so for here, I'm trying to assemble a species of mangrove called, well, let's call it this for now, Avicennia rumphiana. So the closest species in the database to this with an assembled genome is Avicennia marina. Based on genome comparison, these two species diverged about 3 to 4 million years ago, which is a pretty long time. So whether we can use the Avicennia marina genome or not was quite questionable. Okay, so we use a program called ragtag, and I don't think I have the code to run that on this computer. But to validate the output, what I do is run a program called NUCMA, which is a part of the MAMA program. Okay, so you guys should probably keep better records of your stuff than me. What I tend to do is keep a text document in the folder of each project where I make a list of everything that I did, all of the programs that I ran, with some little annotation notes to remind myself of what I did when I come back a few years later and try and figure out what the hell I did, which always happens. Okay, so looks like I've got some code for BWA. This code just here is to get the percent genome identity. I'm calling it AL, that's because this species has a few different names. So we can ignore that bit. Okay, so I've got the contigs of our genome assembly, AL cont. This was scaffolded using ragtag with the no correct option. So the no correct option is important when using ragtag against a different species, because ragtag was originally designed for same species and assembly. So the idea is if you're sequencing a human genome, you expect that contigs, that if you have one contig with sequence from 2 different chromosomes, that it's probably an assembly error. So what the program does is split those contigs and map them independently. So if I'm telling it not to do that, which means that if there is a contig that's composed of sequence from multiple different reference chromosomes, we can map that back and see if we get a large number of those, that maybe the reference that we're using is not appropriate. So here what I've done is I've used NUCMA to map the scaffolds to a marina. And so this is just the NUCMA code. And then I convert that into a coordinates table. I won't rerun everything, it'll take too long. I tend to save all of the stuff that I've done as an R data file. It's faster to load it back in than rerun it. Okay, so the output of assembling this genome was 24,000 contigs. And so I've got a table of the GC content and their length. The largest contig was 2.7 megabases, which is pretty good for a genome assembly. And the shortest contig was 300, which is, you always get a lot of really short contigs. Okay, so the reference that we mapped it to had 32 chromosome level contigs, plus the mitochondria and the chloroplast. Okay, so the first thing that I did was to map our 24,000 contigs. And I did this using a program called BWA. No, that's not what I wanted to do. Oh, actually, we can ignore this line. This is calculating the percent identity. Which is probably a good thing to do if you're using a reference. Calculate the percent identity. So in this case, a total of 68.7% of the A. rumfiana genome could be mapped to A. marina. And of that 68.7% of sequence, it had an average identity of 87.7%. So the overall genome similarity was 60%. So that's about what you get from 4 years of evolutionary divergence. Okay, so I do the plotting of the output file in R, but the program Mama has a plotting function, although since I run Mama on the high performance computer that we have here, it doesn't do the figures as well. So if you're doing it on your computer, you can probably just use Mama's native figure generation. Okay, and so after you've run NUCMA, it essentially generates alignments, and so you can generate a dot plot showing the orientation of all of your contigs that have been scaffolded against the reference sequence. And so if it's an appropriate reference to use, then most of the output will look like this. And you can do this for individual contigs as well. And if it's an inappropriate reference, then you'll get a lot of cases like this. So in this picture right here, you can see that there are two individual large boxes. This means that the program has sequence from two of the reference chromosomes that's been scaffolded into one chromosome from your species. So if you're using a species that is too distant, then you're going to get a single contig that has hits to multiple individual reference chromosomes, and they're probably not even going to be as well ordered as this. I don't have an example to show you of an inappropriate reference though, but you'd see a lot of inversions, you'd see a lot of discontiguous mapping. So instead of seeing a nice line like this, you'd see a lot of random dots all over the place, and it'd be split up between multiple boxes. Yeah, that's about it. Any questions? If not, I think we can end it there.